गुड मॉर्निंग स्टूडेंट्स। आज हम लोग स्टार्ट करने जा रहे हैं चैप्टर नंबर 12 दैट इज़ बायोटेक्नोलॉजी एंड इट एप्लीकेशंस। इन दिस रिगार्ड we have to study about the biotechnology and its application in many fields. As we know that in the biotechnological field, it is found that so many applications and applied examples in case of the food industry, in the chemical industry, in the field of medicine, in the field of agriculture, in the field of power industry, and in the field of environmental protections and environment also, ecology also. So there are so many areas where this branch can be applied. In the first part, we have to learn about the biotechnological applications in the field of agriculture. So today's topic is the biotechnological application in the field of agriculture. If we have to introduce this biotechnology in the field of agriculture, so we know that the biotechnology deals with industrial scale production of biopharmaceuticals and biologicals using the genetically modified microbes or organisms, fungi, plants and animal. And the application of the biotechnology, it includes the therapeutics, the diagnostic techniques, genetically modified crops for in the field of the agriculture, the processed food, bioremediations, waste treatment, as well as in the energy production also. But in the case of the areas that are major areas of the biotechnology is that to supply the best catalyst or the microbes or pure enzyme in form of the improved organism with the better characteristics for the helping of the human being, and to create the optimum conditions for the genetic engineering for a catalyst to do the function. And next is the major research in the field of the purification of that particular protein or the organic compound by developing the downstream technologies. So in the case of the biotechnological application in the field of the agriculture, there is three things we have to achieve: that increase in the food productions by means of the agrochemical based agriculture. Rather than using of the traditional agriculture process, we have to use the agrochemical based agriculture so that we can yield our agricultural product as per requirement of the exploring population. And next is the to develop the organic culture, which is a native culture, and genetically engineered crop based agriculture is also the application in the field of the agriculture. In the field of agriculture, one thinks that we know that their name green revolution, you have heard also that green revolution. What is that green revolution? Green revolution is basically the crop production that increases by using the improved crop varieties, better crop management practice, and use of the agrochemicals, means the fertilizers and pesticides, the adequate amount of the fertilizers and pesticides in the field, farmer field. In the green revolution, we have the food supply was tripled, as we know that it is the present things was not enough for the growing population. We know that day by day our population is going 3 to 4 times in a every 10 years. We find that we are going to double, so that we have to supply the triple food contents for the growing population. Agrochemical were too expensive for the farmers to afford, so uh, we have to make it less expensive for the farmers so that they can afford that value. And increase in the yield with the existing variety are not possible with the conventional breeding. The conventional breeding process is giving a very low yielding varieties, so we have to improve the high yielding variety with the help of the biotechnological processes. Introduction of genetically modified crops was an alternative to the obtain the maximum yield by understanding the genetics of the crop. When we understand the what is the genetic aberrations of the crops, we will make the genetically modified crops or alternative arrangement. And some useful aspect of this GM plant in the agriculture is, number one point is the improvement in nutritional quality. For example, if we are taking rice as a main basic food components, and there is a requirement of vitamin A. So if we add this vitamin A resistant genes in the rice field, so we will give it more nutritional quality in the paddy crop. Better nitrogen fixation is also aspect of the in the field of agriculture, and tolerance to abiotic stress like cold, drought, and salt or heat conditions. These environmental conditions give a very bad effect on our crops. So tolerance to the abiotic stress may be developed by this process. So it is also the aspect of the in the field of the agriculture. Reduce the reliance on the chemical pesticide like pest resistant crops. So reduce the post harvest losses also. The losses due to the bacteria, viruses, rodents, and other microbes also, we have to reduce it, the losses after the harvesting process. Increase efficiency of the mineral usage by plants that prevent the exhaustion of the fertility of soil. If we increase, if we sown crop each and every years, then there is maybe exhaustion of the fertility of soil, so efficiency of minerals may be decreases. So we have to increase it with the help of this aspect also. The production of desired or disease resistant crop will be developed. If we desire that particular crop should be a disease resistant, we have to produce it with the help of this biotechnology. The micropropagation, or we can say it is a tissue culture method for the biomass, it is also used for the energy production. In this picture you can see the top 10 genetically modified food. Soya, a very important food nowadays. We are using that in form of so many products that is made from the soya, and it is soya bean or the soya, corn, tomatoes, flavor saver tomatoes, very rich tomatoes, potatoes is also very good, and cotton seeds also there, pea plants, papayas, canola, so many rice also, paddy field also. So very good genetically modified foods are available with the help of the use of the biotechnology applications. In this picture you can see here the two apple in the hand of a scientist. One apple is the normal apple and another is the GMO, genetically modified. That is a GMO, a very large, huge apple on the hand of the scientist. And in the other picture you can see a very huge cock that is used for human being. So it is also a genetically modified. It will give a lot of flesh for the people to supply the food content. Next thing is that pest resistant plants have to develop with the help of the biotechnology. So in the field of the agriculture, biotechnology is mostly used for the pest, to develop the pest resistant plant. Pest will not attack on that plant. So the development of the pest resistant plant is reduce the use of the pesticides, because we know that there are so many harmful pesticides. If it is going to be enter in our food chain, it will decay or different genetic problems, or the constituents, or the genetic decay, or the DNA interference also. So pesticides should not use too much. So if we want to reduce the pesticides, there should be a development of the pest resistant plant. One such approach that is introduced by the Bt toxin to a target plant. That approach is given the Bt cotton, Bt corn, Bt rice, Bt tomato, potato, soybean, brinjal, ladyfinger. So many crops that are used with the help of this Bt toxin. What is basically the Bt toxin? Bt is obtained from a bacteria that is named as this Bacillus thuringiensis. That's named in it Bt. Bacillus thuringogenesis, and due to presence of this toxin gene in this bacteria, it is known as the Bt toxin. So Bt toxin, if we clone this Bt toxin from the bacteria and introduced into the plants will develop with this toxic gene, and the pest is not going to harvest it, to eat it, the plant parts, and it is a development of the pest resistant plant. Now, what is the mechanism of this Bt toxin and Bt cotton on this plant? Bacillus thuringogenesis, the Bt toxin. Basically, this gene is responsible to produce a certain kind of protein which is toxic in nature, which kills the insect like Lepidopetrans, the tobacco budworm, RB worms, Coleopteron, and these are known as the group is for the beetles, and the Dipterans, that is flies and mosquitoes. So this is the three main group of the insects in which the Bt toxin will get attacked. The Bt toxin gene expresses itself with the protein crystal at a particular phase of the bacteria, that is insecticidal protein. It is known as the insecticidal protein because it is responsible to kill the insects. So Bt toxin exists an inactive protoxin, and on ingestion of it by the insect, when this insect is going to be eat that part of the plant, it is in form of the protoxin or non-toxin, but it will be getting the activated form of the toxin due to the alkaline pH, because in the gut region of the that insect is alkaline pH. So when it get the alkaline pH, it will get the solubilize the crystals, and this toxin bind to the surface of the midgut of the epithelial cells of that insect and create the pores and then swelling and then lysis. So it will get the death of the insect. So insect will be die. The Bt toxin isolated from the bacteria, and it is coded by that gene is known as the cry gene. What is the name of that gene is given by the cry gene, CRY, cry gene, and different types of this cry genes is now incorporated into the crop by the plant depending upon the type of the crop and target pest. It is known as the cry 1 EC, cry 2 AB, cotton bollworm, and cry 1 EB for the corn borer. It is for the corn bollworm and it is used for the corn. Second one is used for the corn borer. This is the corn borer and Bt toxin. Number one picture, you can see the fruit of the cotton, and it is going to be entered here in the maize also. So when this bacteria is extracted, that cry genes cut into the pieces with the help of this DNA extraction and isolation, and then it have to clone in the desired gene and then transform into the cell and tissue culture. When it is going to be incorporated into the plant, will get the breedings and the plant will get that cry gene. In the corn borer process, there is the Bt gene is transferred from the bacillus into the corn, maca, maize. So corn means this is the European corn borer that feed on the corn plant and ingest the protein encoded by that Bt gene. So the Bt protein that penetrate and collapse the cells lining the gut, and the insect dies. So Bt in Bacillus thuringogenesis in active form is sprayed on the crop, eaten by insects. The toxin get activated, and the alkaline pH of the insects will get the swelling of the gut of the insects, and then at last the death of the insect. So it is a biological method with the help of the biotechnology. In the pest resistant plants, along with the Bt toxin, is not toxic to all pests actually. It is not used for the all pest, or pest become resistant against the Bt toxins after some times. So another approach to develop the pest resistant crop is against the nematode parasite. the nematode Meladogine incognita. It is a very dangerous nematode from the Ascahelminthes, and found on cut down the roots of the tobacco plants and decrease the yielding. So this another approach to develop the pest resistant crop against this nematode is the RNA interference, RNA interference. What is this principle of RNA interference to develop pest resistant? It is on the phenomena or principle that silencing translation of the specific messenger RNA means that a particular messenger RNA which is responsible to translate the particular type of protein, it have to be silenced, or the silencing or cutting down this process. So RNA takes place in all eukaryotic organism as a method of the cell defense. Actually, it is found in the all eukaryotic organisms as it is a cell defense mechanism, RNA interference. So the silencing of specific messenger RNA due to complementary double stranded RNA, a type of that RNA molecule to enhance or inhibit the translation of messenger RNA. It binds to messenger RNA and prevent the process of the translation. The source of double stranded RNA or complementary RNA, or we can say the virus RNA, or it is also known as the transposons, the genetic elements which replicate via an RNA intermediate. Transposons is known, the genetic elements which is found over there. So from a messenger RNA, the complementary DNA is prepared in such a way that both strand can transcribe. So complementary DNA is inserted into a vector and finally in host tobacco plant. So in host plant, the complementary DNA will transcribe to form the double stranded messenger RNA, which will be cleaved by the dicer enzyme. The enzyme named as the dicer enzyme to form the siRNA. So what is this dicer? Now the question is, what is this dicer? What is this dicer? So we know that this dicer, also known as endoribonuclease, dicer is known as endoribonuclease or helicase. We can see it is an helicase with the help of the RNase enzyme. It is an enzyme that is found in the human and it is coded by the dicer1 gene. So dicer is responsible to facilitate the activation of the RNA induced silencing complex. This complex is known as the RISC, RNA induced silencing complex, which is very essential for the RNA interference. So if the RISC protein is there, means the RNA induced silencing complex is a multi-protein complex that is found in the cytoplasm. So this in the cytoplasmic RNA induced silencing complex, or the RISC, is contain the double stranded RNA which binding proteins, and including the proteins kinase or RNA activator, another that is known as the PACT, the trans activations response RNA binding protein named the TRBP, trans activation response RNA binding protein, and with the help of the dicer also. That process, in the case of this process, the pre-microRNA is converted into a mature microRNA, that is known as the miRNA, and the target, it going to be target the specific messenger RNA species for the process of the regulation. In case of this vector is used, is known as the Agrobacterium tumefaciens. This vector is used for the genetically engineering to introduce the nematode specific to the gene DNA, and this DNA is used. So nematode specific gene introduced into the host plant, that is known as the transgenic plant, and DNA introduced will produce both sense and antisense RNA in the host cell. Since sense and antisense RNA, both RNA are complementary to each other, this is known as the complementary RNA, and to each other double stranded RNA, this is known as the dsRNA. It initiates the RNAi. When nematode infests the plant, introduction of the nematode genome take place and presence of the double stranded RNA, it silences the specific messenger RNA of nematode by initiation of the RNA interference. So parasite will not able to survive in that host which express the interfering RNAi. and transgenic plant resistant and protect from the parasite. Uh, so we can see that how this risk complex is doing, what how it is functioning complex, what is going to do. So RNA induced silencing complex that is known as the risk protein is a multi-protein complex that incorporated one strand of the small interfering RNA, that is the siRNA, small interfering RNA, siRNA, or we can say it is a microRNA also. So risk uses the si or the small interfering RNA or microRNA as a template for recognizing the complementary messenger RNA. When it find a complementary messenger RNA strand, it activate the RNAs and cleave the many parts of this RNA. So this is the messenger role of this RNAs in the Merodogyne incognita. You can see this picture of this Merodogyne incognita responsible to make root gall systems in many crops fields. Gall-like structure in which it is found, this Merodogyne incognita. The females lay their eggs in these roots, and this function is the nematode. So that cell wall, if it's Cyrid, is going to be introduced inside the cell in the plasma membrane, in the receptor, the feeding tube, with the help of these feeding tubes, and cutting down this roots to enter into insides, and it is going to be used the receptor of this plant by signaling the transduction process, and the messenger RNA is formed to cleave it, so that the particular protein is not going to be formed. Hence in this plant this is not going to be survive. So this is the basic process that is used in the biotechnological field in case of the agricultural production. So in the next class session, we will discuss about the role of this biotechnology in the field of the medicine. Thank you and have a nice day.